Welcome to the Epigenetics Podcast from Active Motif. Join host Dr. Stefan Dillinger for lively discussions with leading epigenetics researchers. Hear about their past experiments, what they're working on now, and what's coming next. You know their papers, now get to know them and discover the stories behind the science.
Hello and welcome to this episode of the Epigenetics Podcast. Today I'm happy to welcome James Hackett from the EMBL in Rome on this show. Please let me briefly introduce you to our audience. You got your PhD from the University of Edinburgh in 2010. You then moved on to do a postdoc at the Gurdon Institute at the University of Cambridge. And since 2016, you are group leader at the EMBL in Rome. A question I like to ask every guest to start off our little podcast is, how did you become interested in biology in the first place and then in pursuing a career in science?
Right. Okay. The origin story.
Yeah.
I don't know. I don't know if mine's that interesting. Let me see now. I don't know how far back to go. I guess when I was young, I was really curious. I was really curious how things work. I was kind of taking apart wind-up radios and just seeing how the bits went together, usually after asking. And so I was curious, and I've always been interested in problem solving. I find it, it's. I remember again being young and just even making up problems. How many Ribena cartons can I fit into this room? And then even going as far as even seeing, was I right or was I close to even being right? Getting a few Ribena cartons. So, always curious how things work and interested to test them. But I guess the key thing for me, if I think back, was probably it's classic in some ways. We're teachers. Between about 14 to 17, I just had really teachers that were really inspiring, that I got on with, and science teachers, of course, this is. And following that, I think I was already, so I had this kind of this interest to understand how things work. I've been orientated towards science. And then when I was, about 16 or 17, I got a job in a pub one summer, and it was very quiet during the day. And I ended up reading a lot of kind of popular science books, Selfish Gene, almost like a whale, Steve Jones. And I just ended up thinking, the genome then is just this fascinating problem that we need to solve. This is about the time the human genome was being sequenced, and understanding how this collection of sequence can convert into molecular phenotypes to developmental phenotypes, and ultimately is affecting whole ecosystems was just a problem I was very attracted to. Yeah. So I ended up, of course, studying it, and the rest is history.
So let's talk about your science that centers around investigating the role of epigenetic mechanisms in genome regulation and developmental reprogramming or programming, with a focus on intergenerational epigenetic inheritance. I want to start in 2012. There, a paper was published with the title "Promoted DNA Methylation Couples Genome Defense Mechanisms to Epigenetic Reprogramming in the Mouse Germline." Can you talk about the study, how you got into that, and what you found?
So this was my PhD. I'd gone to Richard Meehan's lab in Edinburgh, and when I joined the lab, they're working on Xenopus. They'd shown that DNMT1, so the maintenance methyltransferase, if you knock it down with morpholinos, it was affecting the mid-blastular transition. So this is activation of zygotic transcription. But more interestingly, they showed that actually they can rescue that, almost all of it, with a catalytic mutant version. So it wasn't necessarily this loss of DNA methylation that was causing this effect. It may be the protein. So then I was interested in asking, well, is this source, what's the role of this, kind of triggered this question? What's the role of the chromatin modification, or in this case, the DNA modification itself in mammalian context in terms of DNA methylation? And at the time, it wasn't, and I guess it's still not clear exactly, but exactly what DNA methylation was doing in terms of gene regulation was not entirely clear. So we wanted to ask this question: does DNA methylation actually regulate developmentally gene expression? And it was about the time that many DNA methylation maps, genome-wide, the first ones were being produced by the likes of Brian Lister and Alex Meisner and colleagues. And it was becoming clear that most genes were either not methylated, they had CpG islands, so they had many substrate for DNA methylation, which is a CpG dinucleotide. They had many of them in their promoter, but they were always unmethylated, irrespective of expression. And then another group of genes had very low CpG content in their promoter. The substrate was very spread out, and they were often methylated, but the density of methylation was not enough to affect transcription. So it wasn't clear if really any genes were regulated by methylation in a mammalian context. So we came up with this assay whereby we would remove global methylation using 5-aza-deoxycytidine via a chemical approach. And people, of course, have done this many times before. The challenge there is at least many, many indirect effects. Thousands of genes are misexpressed, the vast majority of which are never methylated or regulated by methylation. So our idea was to then remove the drug, to sort of do a hit and run assay, and ask 2 weeks later, now what do we see? On the basis that the stress response, the indirect effects would be rescued, but because there's very little de novo methylation activity in somatic cells, that much of the DNA methylation would not return, and therefore we could identify perhaps DNA methylation gene-dependent genes. So we intersected that, of course, with some genetics and came up with a list of genes, candidate genes that we thought might actually be regulated by DNA methylation. Almost all of them turned out to have methylated weak CpG islands, or intermediate CpG islands. And interestingly, they all had a very similar expression of functional profile, which is that they're expressed almost specifically in the germline, in early germ cells, and I think this was the interesting bit, seemed to be involved in a similar functional pathway. So I guess what we were able to do is show that methylation at this kind of subset of germline-specific genes had a fairly niche but nuanced role in regulating their expression. They became DNA methylated in somatic cells, and in the germline and primordial germ cells, they became demethylated and activated during a global epigenetic reprogramming phase that happens during development.
What you then did is you tried to uncover the mechanism of erasure of this DNA methylation. How did you approach that, and what did you find then?
That's right, yeah. So at the end of my PhD, so we found this group of germline-specific genes, along with other people at the time, it has to be said, that seemed to be predominantly dependent on DNA methylation, at least in somatic cells, for their silencing. And they were involved in genome defense, particularly, as I mentioned. But of course, the next question for me was, well, how are they being demethylated specifically in the germline in order to activate their expression? And around that time, and I guess for the decade or so before, there was emerging evidence from the likes of Petra Haikova, Mitanori Saito, Zimsirani, and many others that the primordial germ cells, the origin of the mammalian germline, are epigenetically fascinating. They go through this wide or genome-wide epigenetic reprogramming event that involves almost complete erasure of DNA methylation, a complete reorganization of the chromatin profiles. And it seemed that these kind of DNA methylation dependent genes were kind of riding on this wave of demethylation to induce their temporal activation. So the question was, how is this working mechanistically? And at that point, I moved to do my postdoc with Azim Surani. And around that time, or a little bit before, the 5-hydroxymethylcytosine and the TET-enzymes had been sort of rediscovered. And the hypothesis, of course, was this may be involved in some way in DNA demethylation through acting as an oxidation intermediate, along with potentially other mechanisms involved. So I went to go and test that question: is 5-hydroxymethylcytosine involved in any way in demethylating? I have to say, specifically germline genes and imprinted loci, or more broadly, and what are the other mechanisms at play?
And what did you find specifically about the mechanisms there?
So
was it the TET enzymes?
Yeah, I mean, I think what we found is that it's a complicated system that involves at least partially redundant mechanisms. So what seems to be happening, and soon after they're specified primordial germ cells, is there's a downregulation of multiple mechanisms, like a transcriptional and a translational downregulation of multiple mechanisms that are involved in maintenance and de novo methylation. So, for example, UHRF1, which is a key partner of DNMT1, is very strongly downregulated, as are the de novo methyltransferases. So very soon after the germline specified, the mechanisms that maintain and establish methylation kind of go down to minimal levels. So this leads to, at the very least, a global passive loss of methylation. But there's probably other things happening at the same time. So, for example, the TET-enzymes, particularly TET1, is upregulated in the germline, and there does seem to be a role, particularly at genomic imprints and methylated CpG islands, ensuring robust demethylation. But what we found is there seems to be a transient enrichment of hydroxymethylation at imprints, certainly relative to the methylation substrate. So as a ratio, there seems to be a faster rate of turnover. And this seems to play at least some role in ensuring complete demethylation, because if you remove TET1, you lead to incomplete erasure of genomic imprints and some delay or failure to activate germline-specific genes. The exact way that has a role, I think, is still an area of investigation. Some very recent papers have shown a role in the human germline during reprogramming just in the last week from Mitanoi Saito. But the TET proteins have both catalytic and non-catalytic roles. They have local effects. And they also seem to directly affect cell identity as well. So I think it's a very complicated process that we still haven't quite got to the bottom of. But what we can say, I think, is that there is both passive DNA demethylation via suppressing the system, the methylation system. The TETs have a role probably in ensuring robustness of locus-specific demethylation, and there may well be other things going on as well.
So it's not only the 5-MC that is important here, but maybe also like the ratio between 5-MC and 5-HMC, and also 5-HMC itself.
Exactly. Yeah. I mean, so, at least in certain cell types, pluripotent stem cells, for example, there's relatively fast turnover of 5-MC via HMC, certainly at certain loci. But of course, the rate of that conversion is important. So, if the 5-HMC levels stay stable, but the substrate is greatly reduced, that effectively means you've got a faster dynamic of turnover. So that's kind of an important consideration as well.
Yeah, we were now talking about the germline, and you just mentioned also the pluripotent stem cells, which you then moved into. So you investigated the role of DNA methylation across distinct pluripotent states and mapped genome-wide 5-MC and 5-HMC in multiple PSCs. Can you talk about this study and what you found?
Yeah, absolutely. I mean, working in a germline is fascinating because it's like watching rejuvenation kind of almost down a microscope. And this is, the reprogramming that occurs in primordial germ cells is just, it's essential for the continuity fundamentally of the species. So a very exciting topic to work on, but also very challenging because there's very few germ cells that you can get your hands on. So doing kind of mechanistic or high throughput studies is, or at least at the time, was fairly challenging. So to do sort of more mechanistic or broader scale questions, I moved partly to a stem cell based models. And in parallel, then there were two projects going on. One is, which you mentioned, which is, are there different states of pluripotency, in vitro pluripotency at least? And what's the underlying functional and epigenomic properties of those different states? So we ended up kind of coming up with 10 different ways of culturing embryonic stem cells and asking what's the difference between them? Does it have a functional impact? And the answer was there is clearly differences. We saw different DNA methylation status. We saw different gene expression profiles, but they were all functionally equivalent at the level of pluripotency. They all were capable of multi-lineage differentiation and self-renewal. But there was, it turns out, some bias in their preference for different lineages. So certain pre-existing pluripotent states would have a tendency to form certain lineage, the neural lineage or the germline lineage, for example, when others might have an opposite tendency. And what we began to find was that maintaining pluripotent cells in different initial media, so with different signaling pathways activated, was resulting in quite substantially different epigenomic profiles, ranging from global hypomethylation to hypermethylation, as well as changes in chromatin states. And this actually had functional implications. So all of the different starting pluripotent states were functionally pluripotent. They could give rise to multi-lineage differentiation. They could self-renew, but they had different biases and different preferences for which lineage they would make more efficiently. So this was interesting for a number of reasons. It started this idea for me that the changes, temporarily uncoupled changes in, for example, the epigenome or genome regulation that actually don't manifest at the time, but can manifest actually at a much later functionally time point. And it was also an opportunity for me to learn new techniques, new skills. I learned a lot of bioinformatics during that project, which has proved to be very useful.
Yeah, you just said and mentioned that you not only looked at DNA methylation, but also at epigenetic states. So what else did you do other than DNA methylation, or what did you look at?
At the time, we were working on a macro level. The focus was really kind of functional, transcriptional, and using DNA methylation as a readout for, let's say, the kind of global epigenetic state. Beyond that, we were just using immunofluorescence to get ideas on Western blots to get global levels of chromatin marks across the pluripotent spectrum.
You also looked at how those states would change in response to varied extrinsic conditions. So what were those extrinsic conditions and what did you find?
Yeah, I guess that's all linked up. So the extrinsic conditions in that scenario are the signaling pathways that are added to the culture media. So, for example, when you are culturing naive pluripotent stem cells, at least mouse, you're typically activating Wnt signaling into eyeless media through the addition of chiron, or inhibiting MEK through using PD. And by kind of changing the relative, either the presence or the relative concentration of various inhibitors or additives or signaling molecules added to the media, you're changing the kind of extrinsic cues that the cells are experiencing. And of course, this is propagating to the genome and epigenome level.
Yeah, you just mentioned that you were working on other approaches to look at the same thing. And then you developed a CRISPR screening to investigate cell fate transitions. Was that something that you were just referring to, and what did you find using this approach?
Yeah, that was a really nice project. That was an organic project. So of course CRISPR came out, and everyone was very excited by the idea. This is going back 2012, 2013. Very excited by the idea that you can now make precision perturbations, and you can do it pretty quick. And so I shelved that for a year, that concept. And then I started to think, what I would really like to do is not be able to just make these precision changes and knock out rapidly, but to do it at scale. How can we functionally interrogate hundreds, thousands, or tens of thousands of genes at the same time using CRISPR? And I started to think about whether we could screen using this approach. And of course, with probably most of my ideas, I thought it was a fabulously novel and great idea, but many people had the idea before me. And I started to reach out to people, in this case, Koseki Yusa, who was based at the Sanger in Cambridge, who'd been developing a CRISPR screen approach very soon after CRISPR turned up. And I was excited by the idea that we might be able to ask a very long-standing question in the field, which is, what's the origin of the germline? In other words, what's the genetic factors required to generate the mammalian germline lineage? And this is a very interesting question because mammals use an inductive mechanism of specifying their germline. So during early development, embryonic cells are primed or fated to become somatic lineages. And the germline is actually redirected from that fate and specified through activation of key transcription factors, which then initiate a cascade that includes epigenetic reprogramming that we discussed earlier. But how that works, how that specification of the germline works, was not at all clear. People had used, and it's still not entirely solved, people had used mainly a candidate approaches, looking for genes that are expressed in the early germline, and inferred from that they might be functionally relevant. And in many cases, that was true. Genes like PRDM1, PRDM14, or BLIMP1, PRDM1 had been discovered that way. But doing this in an unbiased and systematic genome way was completely not tractable because only, at least in vivo, 30 or 40 cells, primordial germ cells, are specified. So I had this dual idea of being able to use or exploit a CRISPR screen, which enabled sort of unbiased functional questions on a genome scale. But then we needed to be able to make germ cells at a very large scale in order to do that. And about the same time, Katsuhiko Hayashi and Mitsunori Saito were developing an in vitro model of primordial germ cell specification. And so these two things just came together perfectly. And we could now ask the question, if we knock out every gene in the genome, firstly, how's that involved in transition first from a naive pluripotent state to a formative state and ultimately to a germline state using this in vitro model? And that proved to be, very challenging, but also very informative. We were able to identify many of the genes that were known, so BLIMP1, PRDM14, AP2 gamma, these kind of things came up. But also then this wonderful unbiased candidate list of functionally, well, not necessarily validated, but genes that were assessed based on their functional role in deriving in vitro germ cells. So this was it was interesting biologically, but it was also an awakening for me that you can do both precision perturbations, so specific knockouts, and you can do it at scale, so you can test the whole genome at once. And this struck me as an extremely powerful approach, in this case, to understand regulators of the germline and ultimately reprogramming after. But of course, for any question for which you can design a good assay and a good functional readout or phenotypic readout.
Yeah, I just wanted to mention that this sounds really like a lot of work if you're knocking out like every factor or at least many factors.
Well, that's the beauty of pooled CRISPR screening. You effectively have one well, and within that well, every cell is knocked out or at least, depending on your screen design, knocked down or activated for a specific gene. So you have this incredible power. But compared to an arrayed or a candidate approach, a lot less tractability, a lot more tractability.
And last but not least, you looked at epigenetic inheritance. I also found a publication where you looked at a specific factor. Maybe you can talk about that for a minute.
That's right. So once I established my lab, which I think is what you're referring to, there were two broad questions. One was sort of really trying to develop tools to ask mechanistic questions at the molecular level about what chromatin modifications, what DNA methylation is actually doing in a very locus and context-dependent way, with sort of the ability to understand quantitative relationships. So that's very molecular. But the much broader question that was done under the same umbrella of epigenetics is, do any potential changes in epigenetic or chromatin states that may occur in the germline, do they have the potential not only to transit through mitosis, but also potentially meiosis and influence early development? So those were the two questions. And the idea was that if we built molecular tools that could make specific changes in chromatin modifications, we could actually, of course, test their direct functional role, but also test their effects on or their ability to propagate or perhaps transmit through mitosis and meiosis. So that was the foundation of what we wanted to do.
Yeah, go ahead and talking about the results. Yeah, exactly.
Yeah, so I'm not sure which one you're referring to exactly, but the first kind of study we did actually exploited CRISPR screening as well. I was set up to do that, so I knew we could do it. And we were interested in revisiting this question. What's the mechanistic basis of global epigenetic reprogramming events, and particularly DNA demethylation events? So to test that, we first exploited a reporter system of DNA methylation so that we could have a live readout, a single cell readout of the methylation states that we could track temporally. This was built on a reporter system initially built by Rudy Janisch and colleagues. We adapted it slightly. It's a very, very elegant system that uses an imprinted promoter to drive expression. So imprinted promoters will not acquire de novo methylation aberrantly. They're resistant to that, but they are sensitive to methylation. And to exploit that secondary aspect, you hook it up in cis to an element that acquires the methylation state or an average methylation state that's average for the genome. Because it's adjacent in cis, it actually just transfers that methylation state to the imprinted promoter, which in a kind of linear or a predictable manner changes the expression. So you now have a reporter of global DNA methylation. You can use in vitro models to mimic both the loss of or erasure of methylation and the acquisition, the developmental acquisition. So you can now transit between high and low methylation states. You can record that at single cell live. And then if you screen on top of that knockout screen, you can ask, what do I need to remove to prevent global DNA methylation? And what do I need to remove to prevent acquisition? And so that's what we did. And we identified a number of factors that seem when you remove them or perturb them, they strongly affect methylation. Again, many of the positive controls, many novel factors, for example, involved in RNA modifications. They seem to be influencing DNA methylation levels. But very interestingly, almost all of them validated, 18 out of 20 top hits validated in terms they were having at least some effect on the ability to demethylate, ranging from partial to significant. But the very top 2 hits were having no effect on global methylation, but they were the strongest hits by a mile. And these were 2 proteins called DPPA2 and DPPA4. They're very interesting proteins. They're mitotic bookmarking factors, so they stay bound to chromatin during meiosis, and they seem to be involved in targeting H3K4 trimethylation. And it turns out they weren't affecting global levels, but they were very much affecting focal DNA methylation levels. So they're found bound at almost every single CpG island promoter, almost all promoters in the genome, where they seem to be involved in preventing acquisition of de novo DNA methylation. In other words, to maintain their permissiveness for activation much later in development by preventing their long-term silencing. So the take-home message of that study was that, yes, we identified factors involved in global events, but also complementary factors that are involved in ensuring that the methylation state of specific loci is maintained in an appropriate state during both the kind of programming and reprogramming phases.
Mm. And I said last but not least to my last question, but just 2 weeks ago there was, and we are recording this in May 2024, there was a paper in Nature Genetics, and I just found this yesterday, titled "Systematic Epigenome Editing Captures the Context-Dependent Instructive Function of Chromatin Modifications." And there was quite a buzz about this paper. Can you quickly talk about the study?
Yeah, absolutely. So again, this was a goal since setting up the lab 6 or 7 years ago, was to build tools that enables us to make very precise changes in the epigenome so we can really go back and answer some of these quite long-standing cause and consequence questions. So we've known, because of the efforts of many consortia and labs, that exactly where most chromatin modifications are in the genomes, across tissues, across cell types, and so forth, across disease. And we correlated this, of course, with transcription and many other DNA templated processes and genomic variation to infer correlations between the presence of a chromatin modification and a certain output. But to kind of get at causal relationships, typically there's a perturbation involved. And by and large, the field has taken the approach of interfering with one of the chromatin modifying enzymes. Used to be typically by knockout, more recently perhaps by degron or specific inhibitors or using an oncogene. And this is a very powerful way of perhaps of interfering with chromatin marks to ask, what are they doing functionally? The challenge is that these perturbations typically affect the entire genome simultaneously, and therefore you get widespread indirect effects. Possibly the majority of the molecular and developmental phenotypes we're seeing are indirect effects rather than direct consequence of loss of a chromatin mark. And many of these enzymes also have very important non-catalytic roles. They're involved in many complexes, which often fall apart when you interfere with the protein. So actually, it's quite difficult to interpret exactly what chromatin modifications are doing simply by interfering with the key enzyme. So what this means is actually there's some quite fundamental questions that remain outstanding. One is, when you see a certain chromatin mark and it's associated with a certain transcriptional stage, is that a cause or a consequence, or perhaps somewhere in the middle? And we also have relatively little understanding about context dependency. So when we do let's say an epigenome profile, we get 10,000 peaks. They're all peaks, but we don't necessarily have the knowledge to interrogate how does the underlying, or each of those pieces will have a different underlying DNA sequence and a different epigenomic context and perhaps different transcription factors are binding, and we don't really have the understanding to fully understanding what the interaction is between that chromatin mark and the context-dependent factors underneath it, or the quantitative impact of that mark, instead of perhaps being an on-off toggle for expression or some other DNA templated processes, it's just having a much more subtle effect, tuning the system. So our idea was to get at these questions. It's not just our idea. There are many others in the field that are developing epigenome editing tools, but that we should be able to go in and make a very precise change at a specific locus where we know exactly what we've done, and then we can read out sort of the causal effects. So we built a toolkit. It's based on a dead Cas9 that, of course, binds but does not cut DNA. It's linked with a modified Sontag tail with sort of large spaces that enable recruitment. In this case, specifically the catalytic domain of a chromatin modifier. So we don't want to recruit the entire chromatin modifier. We don't want to recruit complexes. We want to specifically recruit the catalytic activity to ask what's the function of the mark. So the system enables epigenetic editing of 9 of perhaps the more prominent chromatin modifications, so H3K4 methylation, K27 methylation, K119 ubiquitination, and so forth. And it works pretty well. We can deposit pretty high levels, physiological levels of chromatin marks at target loci, and off-targeting is pretty minimized. And so using this tool, we systematically went through, we programmed each mark, we programmed it to the same DNA sequence, but in different kind of expression states. So we could ask, how does one mark affect gene expression starting from different states without underlying changes in DNA sequence? So we really took a reductionist approach. And we built a catalytic point mutant for every effector. So we could ask, okay, is it really the mark that's doing it, or is it recruitment to the system? And the take-home message of what we found is, firstly, measured at a single cell and quantitative level using reporter systems, pretty much every single mark has at least some detectable effect in at least one or two contexts. This could be very subtle. Perhaps it only affects a small fraction of cells and relatively small effect size. But in many cases, it was a very significant effect. It was instructively driving either repression or activation or upregulation, I'd say, of transcription. That was the first key thing we found. So we were able to quantify at least within certain or tested contexts, the actual direct effect of each chromatin modification. And then we asked the question, how does that interact with the underlying DNA sequence? So we started systematically making changes in the underlying DNA sequence to ask, what's the interaction between the starting underlying motifs or variants and then the mark on top? And then by and large, we found the effects were coherent. So H3K9, for example, would drive transcriptional repression pretty much irrespective of the underlying DNA motifs or variants, but there were clearly quantitative relationships there. So in that case, for example, if we inserted a YY1 motif, the effect of the chromatin mark was greatly diminished. So there's a functional interaction. And this actually led all the way to switch-like functionalities. So for example, if we edited H3K36 trimethylation at a promoter, it pretty much had mixed or very subtle effects in almost all circumstances. But in the presence of a CTCF motif, it switched behaviors and now went from being neutral to a very powerful transcriptional repressing mark. So the take-home message there is really the function, the transcriptional function of a chromatin modification is not unequivocal, but very, very context dependent and dependent on a number of factors, including the underlying DNA sequence, transcription factors, and almost certainly the underlying or the cell type in which that mark is being programmed.
Imagine you're due to submitting a grant proposal tomorrow, and you maybe are. What would you have written into that?
A grant proposal tomorrow. I mean, listen, I don't know if I want to go through my grant so that people will lose the will to live if I went through what I'm applying at the moment. But I can tell you, what I think is really exciting going forward is making, and I've alluded to it in the last discussion, that being able to make increasingly precise perturbations, I think really is an important part of our future. Over the last few decades, I think the community has really expanded our ability to read out at higher resolution and in a much more precise way cellular phenotypes, molecular effects. Now we have spatial, single cell, multi-omics, we really can get down. But the ability to perturb the system up front perhaps hasn't progressed in quite the same rate, so ability to read out the effect. So I'm really excited about the ability to make precise changes either to the epigenome, for example, through epigenetic editing, and the genome through creative uses or highly scaled uses of CRISPR, to really dig into cause-consequence relationships and actually the complexity, the context-dependent complexity in the genome. So these are big picture questions I'm interested in. And we didn't touch on it so much, but also how the environment may play into that, how environmental contexts are influencing cellular identity, organismal phenotype, and potentially even the germline and beyond.
Yeah. So for the last 37 minutes, we have been on a journey through your scientific career. You just mentioned something that we missed, but did we miss something important, or would you like to add something?
Yeah, there are a few things. We just in parallel, I mentioned it at the end, we've been asking, how does the environment actually influence genomic readouts or epigenomic states or some other macromolecules within the germline? And our recent work has really investigated the communication using the gut microbiome as a paradigm of environmental change, communication between that and the germline. And we're very excited to understand how and what changes might occur, epigenomic changes might occur in the germline, and how that may influence offspring phenotype. We've recently reported on that. And I think another point is, we talked a little bit about some of the work I've been involved in, but all of that is being done in collaboration, either with mentors and colleagues that have supported me and that we've kind of brainstormed together, or more recently by the people in the lab. And I just want to point out that this stuff is not done without them, and they've done just really incredible work that's kind of helped me on my journey.
So thank you, James, for your time and for being on the show.
Thanks very much, Stefan.
Thanks for listening to this episode of the Epigenetics Podcast from Active Motif. We hope you enjoyed it. You can find all the mentioned references in the show notes. Please rate, review, and subscribe to our podcast on your favorite podcast platform so you never miss an episode. We'd love to hear from you, so please send us your feedback on Twitter, Facebook, LinkedIn, or via email at podcast@activemotif.com, and we'll give you a shout out in a future episode. For more great epigenetics content, check out the Active Motif blog at activemotif.com/blog. Thanks for listening, and stay tuned.